Humans produce many types of chemicals that end up in the environment. In order for these chemicals to be authorized, regulators must ensure that they have no negative effects on animals and plants, and consequently for humans. To do so, chemicals are tested using fish, leading to the sacrifice of more than 3 million fish each year. We at Eawag have a mission: to replace testing with fish by non-animal methods that we develop using permanent culture of fish cells, also called fish cell lines, which are initiated from only one single donor fish. Following our mission, we have developed the RTGIL-W1 cell line assay to replace acute fish toxicity testing. This test has been validated and received global acceptance by both ISO in 2019 and by the OECD in 2021. With this important step, we have demonstrated that it can be done, that fish cells can replace ethically questionable fish tests. Currently, many of our activities focus on expanding the portfolio of cell line-based assays, such as in our NFP project, where Jessica is developing an assay for evaluating the effect of neuroactive chemicals on fish brains, while Mihai is studying how proteins can be used as molecular markers of toxicity. However, in our experience, moving this assay from research into practice is not easy, because industry and regulators tend to remain skeptical about these new methods. To address that problem, our NFP project seeks to put the scientists and the end users on the same boat from the beginning and to co-design these new methods together. We therefore have joined forces with social scientists experienced in innovation processes to develop the new methods together with our industry partners and with the regulators. This is our solution: bring together different stakeholders from the start to pull on the same rope and move alternatives to animal testing over the finishing line.